Plant Growth and Development Part 1. In Chapter 5, you have already studied the organization of a flowering plant. Have you ever thought about in an orderly sequence where and how the structures arise like roots, stems, leaves, flowers, fruits, and seeds? By now you are aware of the terms seed, seedling, plantlet, mature plant. You have also seen that over a period of time, trees continue to increase in girth and height. However, not only the fruits, flowers, and leaves of the same tree have limited dimensions, but also fall and appear periodically and sometimes repeatedly. In a plant, why does vegetative phase precede flowering? All the organs of a plant are made up of a variety of tissues. Is there any relationship between the function they perform and the structure of a cell, a tissue, and an organ? Can the function and the structure of these be altered? All the plant cells are descendants of the zygote. Then, how and why do they have different structural and functional attributes is the question. The sum of the two processes is development, growth, and differentiation. To begin with, it is essential and sufficient to know that from a zygote, which is a fertilized egg, the mature plant development follow a succession of events which is highly ordered and precise. A complex organization of the body is formed during this process that produces roots, leaves, branches, flowers, fruits, and seeds, and eventually they die. In this chapter, you will also study some factors which govern and control these developmental processes. These factors of the plant are both intrinsic and extrinsic. Growth. One of the most fundamental and conspicuous characteristics of a living being is regarded as growth. What is growth? A permanent irreversible increase in size of an organ or its parts or even of an individual cell can be defined as growth. Growth generally is accompanied by metabolic processes that occur at the expense of energy. Therefore, for example, growth is expansion of a leaf. The swelling of a piece of wood when placed in water, how would you describe this? Plant growth generally is indeterminate. Plants retain the capacity for unlimited growth throughout their life as plant growth is unique. This ability of the plants is because meristems are present at certain locations in their body. The cells present in such meristems have the capacity to self-perpetuate and divide. However, such cells make up the plant body as they soon lose their capacity to divide. This form of growth is called the open form of growth, wherein by the activity of the meristem, new cells are always being added to the plant body. If the meristem ceases to divide, what would happen? Does this ever happen? You have studied about the root apical meristem and the shoot apical meristem in chapter 6. You know that they contribute to the primary growth of the plants and also cause the elongation of the plants along their axis. You also know that the lateral meristems, vascular cambium and cork cambium, appear later in life in dicotyledonous plants and gymnosperms. The increase in the girth of the organs in which they are active is because of the meristems. This is known as secondary growth of the plant. Growth is measurable. A consequence of increase in the amount of protoplasm is principally growth at a cellular level. Since it is difficult to measure directly increase in protoplasm, one generally measures some quantity which is more or less proportional to it. Therefore, by a variety of parameters, growth is measured, some of which include cell number, increase in fresh weight, dry weight, area, length, and volume. You may find it amazing to know that one single root apical meristem of maize can give rise to 17,500 new cells or more per hour, whereas the cells may increase in size by up to 350,000 times in a watermelon. Growth is expressed in the former as increase in cell number, whereas growth in the latter is expressed as increase in size of the cell. While in terms of its length, the growth of the pollen tube is measured, while in a dorsiventral leaf, the growth denotes an increase in surface area. Phases of growth. Generally, into three phases the growth period is divided, namely meristematic, elongation, and maturation. By looking at the root tips, let us understand this. Both at the root apex and the shoot apex, the constantly dividing cells represent the meristematic phase of growth. In this region, the cells are rich in protoplasm and they possess a large conspicuous nucleus. Their cell walls are thin, cellulosic, and primary in nature with abundant plasmodesmatal connections. The cells proximal, that is, just next away from the tip to the meristematic zone, represent the phase of elongation. In this phase, the characteristic of the cells are cell enlargement, increased vacuolation, and new cell wall deposition. More proximal to the phase of elongation, that is, further away from the apex, lies the portion of axis which is undergoing the phase of maturation. In this zone, the cells attain their maximal size in terms of wall thickening and protoplasmic modifications. In Chapter 6, most of the tissues and cell types you have studied represent this phase. Growth rates. Growth rate is the increased growth per unit time. Thus, mathematically, we can express the rate of growth. In a variety of ways, an organism or a part of the organism can produce more cells. The growth rate shows an increase that may be arithmetic or geometrical. Following mitotic cell division in arithmetic growth, only one daughter cell continues to divide while the other differentiates and matures. By a root elongating at a constant rate, the simplest expression of arithmetic growth is exemplified. In the figure, a linear curve is obtained on plotting the length of the organ against time. It is expressed mathematically as. In the geometrical growth, let us now see what happens. The initial growth is slow in most systems and thereafter it increases rapidly at an exponential rate. Here, following mitotic cell division, both the progeny cells retain the ability to divide and continue to do so. However, the growth slows down with limited nutrient supply, leading to a stationary phase. We get a typical sigmoid or S-curve if we plot the parameter of growth against time. Growing in a natural environment, a sigmoid curve is a characteristic of living organism. It is typical for all cells, tissues, and organs of a plant. Can you think of more similar examples? In a tree showing seasonal activities, what kind of a curve can you expect? The exponential growth can be expressed as follows. Here, the relative growth rate is R, and it is also the measure of the ability of the plant to produce new plant material, referred to as efficiency index. Hence, the final size of W1 depends on the initial size W0. Between the growth of living system, quantitative comparisons can also be made in two ways. Absolute growth rate is the measurement and the comparison of total growth per unit time. Expressed on a common basis, the growth of the given system per unit time, example per unit initial parameter, is called the relative growth rate. Two leaves are drawn in the figure that are of different sizes A and B, but shows absolute increase in area in the given time to give leaves A1 and B1. However, much higher relative growth rate is shown by one of them. Which one and why? Conditions for growth. What conditions do you think are necessary for growth? This list of very essential elements for growth may have water, oxygen, and nutrients. The plant cells by cell enlargement grow in size, which in turn requires water. Turgidity of cells help in extension growth. Thus, intimately, growth and further development is linked to the water status of the plant. The medium for enzymatic activities needed for growth is provided by water. Metabolic energy essential for growth activities is released with the help of oxygen. For the synthesis of protoplasm, nutrients are required by plants and they also act as a source of energy. In addition, an optimum temperature range every plant organism has which is best suited for its growth. From this range, any deviation could be detrimental to its survival. Light and gravity, that are the environmental signals, also affect certain phases or stages of growth. Differentiation, dedifferentiation, and redifferentiation. The cells derived from cambium, root, and shoot apical meristem differentiate and mature to perform specific functions. This act leading to maturation is termed as differentiation. Both in their cell walls and protoplasm, during differentiation, cells undergo few to major structural changes. For example, the cells would lose their protoplasm to form a trachary element. A very strong elastic lignocellulosic secondary cell walls also develops in them to carry water to long distances even under extreme tension. The various anatomical features you encounter in plants try to correlate to the function they perform. Another interesting phenomenon is shown by plants. Under certain conditions, the living differentiated cells that by now have lost the capacity to divide can regain the capacity of division. This phenomenon is termed as de-differentiation. For example, formation of meristems from fully differentiated parenchyma cells, intervesicular cambium and cork cambium. Such meristems or tissues, while doing so, are able to divide and produce cells that once again lose the capacity to divide but measure to perform specific functions, that is, get redifferentiated. In a woody dicotyledonous plant, list some of the tissues that are the products of redifferentiation. How would you describe a tumor? During plant tissue culture, what would you call the parenchyma cells that are made to divide under controlled laboratory conditions? Recall, we have mentioned earlier that the growth in plants can be indeterminate or determinate, that is, the growth is open. Now, we may say that even in plants, differentiation is open because at maturity, cells or tissues arising out of the same meristem have different structures. The final structure at maturity and by the location of the cell within the cell or tissue is also determined. For example, from root apical meristem, cells positioned away differentiate as root cap cells, while those pushed to the periphery mature as epidermis. Development. Development is a term from germination of the seed to senescence that includes all changes that an organism goes through during its life cycle. In the figure, diagrammatic representation of the sequence of processes is given which constitute the development of a cell of a higher plant. It is applicable also to tissues or organs. Plants follow different pathways to form different kinds of structures in response to environment or phases of life. Example, heterophyly in cotton, coriander and larkspur. This ability is called plasticity. From those in mature plants, the leaves of the juvenile plant are different in shape. Due to environment, on the other hand, difference in shapes of leaves produced in air and those produced in water, example in buttercup, also represent the heterophilous development. An example of plasticity is the phenomenon of heterophyly. In the life of a plant, very closely related events are growth, differentiation, and development. Broadly, the sum of growth and differentiation is considered as development. In plants, development, that is both growth and differentiation, is under the control of intrinsic and extrinsic factors. The intrinsic factors includes both intracellular or intercellular factors, while the extrinsic factors includes light, temperature, water, oxygen, nutrition, etc. Thank you so much for watching. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.